Yeah. Okay, well, welcome, folks. We're going to start our evening presentation. If you don't know who I am, my name is Peter Heidi, and I'm here on behalf of Malama Kai Foundation. We have the Malama Puna Oke Kai, or we have thoughtful stewardship of our coastal waters. And as you know or may not know, Malama Kai has been around for a long time. Malama Kai was established in 1991 to enhance marine and coastal protection in Hawaii through education, outreach, and support of community-based resource management for current and future generations. Some of our big highlight programs are the Dave's mooring buoys. In fact, the first buoys were installed in 1990, and now we have over 200 that exist statewide. And I'm sure some of you have used these buoys before. I like to say it's one of the easiest ways you can protect coral. Use a buoy. And Ridge to Reef Stewardship Program, which I work with as well, and that's taking middle schools and taking them to place-based stewardship projects, getting their hands dirty. We're outplanting native species, we're removing invasives, and then of course the ReefTalks and other past projects. We are not-for-profit, so please, if you can donate and also follow us on social media. These Reef Talks are monthly community marine science presentations, started over 20 years ago. Some of you have been here long enough to know that I worked with Sarah Peck with UHC grant in these Reef Talks. And usually on the third or fourth Thursday of the month, funded by donations and hosted by ASU. You may have noticed this amazing coral land-based nursery out here, and Terraformation. So those are the two organizations that are giving us the space for these once-a-month Reef Talks. And with that, I'm going to go ahead and introduce Dr. Grace Klinges, and Restoring with Resilience, Enhancing Coral Restoration, Outcomes in the Wake of Climate Change. So this is one of our fellow researchers with ASU, and enjoy the show.
Try not to block my own slides too much. Thank you, Kater, for the introduction. I'm Grace. This is my office. I sit right there. So it's fun to be up in front of you guys talking and sharing this space a little bit and getting to talk about what I'm passionate about. Oops, I'm going backwards. So I'm going to talk to you today about primarily my work in Florida and how we are bringing efforts in coral restoration that we've performed in Florida to West Hawaii to restore our reefs here. I'm going to start off by telling you guys something you probably already know working here, or living here rather. Coral reefs are one of the most biologically diverse ecosystems in the world. And coral reefs are important for many reasons on top of being biologically diverse. They help protect our shorelines by dissipating energy from wave action. So it helps prevent our shorelines from being battered by waves. And corals and their associated microbial communities produce antibiotics and compounds that are not found in terrestrial ecosystems. And these compounds can be useful for antibiotics or developing medicines to fight disease. They provide essential fish habitat. We know that a lot of our subsistence fisheries depend on our reef communities. Of course, they provide tourism. And at least 1 billion people worldwide rely on reefs for food, coastal protection, cultural practices, and income. What is a coral? Sometimes some of us see these every day, and some people don't know that these are actually animals. Each coral is it's comprised of thousands of genetically identical polyps. So each one of these is a laser, yeah. So each of these polyps is kind of like a tiny anemone. And each of these polyps that makes up the coral is genetically identical. So they're all clonal polyps building this giant meta-organism. And these polyps harbor algae, these photosynthetic algae shown here. And these algae, they photosynthesize and provide food for the coral. They take in sunlight and turn it into nutrition that helps sustain those corals. And they also give coral their color. That's why many of the corals that we see look brown or green. But some algae can make the coral look with beautiful colors like pink and purple that we occasionally see. And the US has over 1,900 miles of reef, including the US territories and the outer islands. And today I'm just going to talk about two states that have coral reefs, Florida and Hawaii. And until this January, I was a postdoctoral researcher at a coral restoration facility in the Florida Keys called Mote Marine Laboratory. And my research there centered on coral resilience and genetic diversity. I recently joined the ʻākoʻakoʻa program here at the Reef Restoration Center. I joined in January, so I'm still really new to the program and to Hawaii. And here I'm helping to lead our restoration strategy and develop research programs for West Hawaii. We're very much in the planning stages. So like I said, I'll be talking a lot about what we did in Florida and how we can apply those principles to restoration here in Hawaii. There's our logos. Coral reefs in Florida are essential to the economy and culture of each state, Florida and Hawaii. In Florida, there's over 350 linear miles of reef. And the direct economic value of this reef is immense in terms of tourism and fish habitat. It's $8.5 billion. It supports loads of jobs, and it brings in over 16 million visitors per year for the tourism industry. It also provides extensive shoreline protection. The Florida Keys are a calcium carbonate reef. They're not basalt like our island here, so they're very easily degraded. And the value of infrastructure that and economic value that these reefs protect is over $675 million per year. In Hawaii, we have over 60% of American coral reefs are in Hawaii alone. And that's over 140,000 acres of coral reef habitat. It's immense. We have a huge economic value of these reefs here. The tourism industry is over $6.5 billion per year, and our fishery value is $3.2 million. And again, an immense amount of infrastructure protected by our reefs, even though we have our islands are basalt. Our reefs are protecting coastal infrastructure in low-lying areas such as Maui. And Hawaii's reefs are so important because of the number of unique endemic species that they harbor. Over 1,200 unique species are unique to the Hawaiian Islands. But these numbers don't come close to stating the cultural, spiritual, and pragmatic importance of coral reefs to the Hawaiian community. In the Kumulipo, the coral polyp is the first organism that came to be. I'm really new here, so I'm still trying to understand the importance of reefs. I'm learning more and more every day about how important these reefs are to the Hawaiian community. But as residents of West Hawaii, we all rely on reefs in ways that we probably don't even realize. We rely on reefs for our food, for the sandy beaches that are made of degraded coral skeletons. Many of us rely on reefs for our jobs, whether you're scientists or in the tourism industry. We use them for surf. But how do you put a number on that value? How do we value the loss of our reef ecosystem? In Florida, the residents have had to face the unfortunate reality of that scenario, trying to put a value on the loss of their reefs. There's been a dramatic decline in coral cover in Florida since the 1980s due to many different stressors, including disease and thermal stress, which causes coral bleaching. It's tragic, and it's resulted in the massive degradation of Florida's coral reefs. So we went from a vibrant reef ecosystem before the 80s to essentially reef rubble. These stressors, as I said, have led to a dramatic decline, especially in the two primary reef-building species in Florida, elkhorn and staghorn coral. And I'm going to talk a little bit about these species today, but for context, Hawaii is also experiencing these same stressors. Hawaii's coral reefs are constantly exposed to numerous stressors, including overfishing, sewage and sediment input, huge storms, as well as thermal stress resulting from climate change. And in some places, these stressors have reduced the otherwise vibrant reef to rubble. So this is a picture that we took last weekend in Honolulu. There's areas where storm damage and reduced coral colonization and just degradation of the coral reef there has turned the reef to rubble, which makes it very hard for new coral to recruit on those reefs. I talked a little bit about ocean warming, and ocean warming is really detrimental to coral because it disrupts that coral-algal symbiosis that I talked about. The coral, under thermal stress, the algae starts to produce free radicals. You guys might see, you know that free radicals are bad because your skincare prevents free radicals. These are just loose oxygen species bouncing around, and they cause oxidative stress to the coral. And during thermal stress, that algae starts overproducing those compounds, and the coral literally evacuates that algae. But because that algal symbiont is providing so much of the nutrition for the coral, that coral starts to starve. So prolonged bleaching, as we see here, that's that stark white coral that lacks algae. Those corals will start to starve and eventually will die. During my PhD, I worked in French Polynesia, and I saw bleaching turn this vibrant reef into a coral graveyard. It was a wasteland of skeleton. And this is the reality of our coral reefs without intervention. We have to work to prevent climate change and increase the resilience of our reefs. I swear the point of my talk is not to make you guys miserable. I am going to talk about some beacons of hope, but I want to start with the reality of the situation that we're facing. The year 2015 was marked as the warmest on record in Hawaii. It led to a widespread severe bleaching event, causing an over 50% loss in coral cover in the region. And then the islands were walloped again with a bleaching event in 2019. And you can see the temperature profiles from 2019 in this map. And unfortunately, those high temperatures did lead to massive amounts of mortality, again, on that already damaged reef. We saw heavy losses in West Hawaii in 2019, and these red colors indicate higher mortality. So more coral died in these areas. So you can see in this area south of Kona, saw heavy losses resulting from that bleaching event. Thankfully, the corals that were most heavily influenced were our cauliflower corals, Passalopora, the genus Passalopora. And these are fast-growing corals, so they were very strongly influenced by the bleaching event, leading many of our healthy corals to look like this. They lost their photosynthetic algae and then eventually died and became overgrown with bad algae, macroalgae. So that is no longer a living coral. That is a coral that is, whoa, sorry. That is a coral that will eventually disintegrate. It's no longer creating new skeleton and it will start to collapse. So the good news is that this species, because the species grows so quickly, it has been able to bounce back to an extent from these bleaching events. The problem is as climate change begins to worsen, we will see more and more frequent bleaching events, and the reef just can't keep up. It can't grow enough new corals quickly enough. And unfortunately, that's not the only stressor we have to contend with here in West Hawaii. This is a crown of thorns starfish. I know the Division of Aquatic Resources is always on top of it, trying to make sure that we're not seeing too many of these starfish on our reefs. It's not an invasive species. It is native to the Pacific, but it can get out of control quickly, and it can be hard to eradicate. Here you can see it literally consuming a Passalopora head. It just is non-specific and it eats everything. I'm just going to give up on this because I keep clicking forward. We also have to contend with anchor damage and boat groundings, as well as planes. Something new this year. This is in Kaneohe Bay. This was a naval plane, supposedly a spy plane that was grounded in Kaneohe Bay. And you can see it literally sitting on top of a field of Parietes compressa finger coral. I believe this is the last sad picture. So it's over. But we have a lot of stressors going on the reef in the ocean, but the inputs of our activities on land also affect the reef. So because of the structure of our island with how steep it is, sediment, nutrients, and potentially harmful chemicals, just pharmaceuticals, things that are produced from industrial activity, these are all moving from Mauka to Makai with the movement of drainage. So the net result of these stressors is that coral cover is projected to decline in Hawaii and Florida over the next century. It's a pretty grim outlook, projected to be less than 1% coral cover here by the year 2100 if we do not take significant action. In Hawaii, we have more time than in Florida, but we have to take action now, both to actively restore the reef and to work to reduce those stressors that reefs experience. So there are lots of ways you can engage in coral restoration. I work by putting coral back on the reef, but you can all work by living a greener lifestyle and engaging with your community and your government to make beneficial decisions for the reef. I engage in active restoration efforts here at the ʻākoʻakoʻa program, and as I mentioned, we are very much in the planning stages, but we intend to engage in active restoration, which entails helping the corals grow by fragmenting them or moving them from parts of the reef that are in bad shape to moving them from parts of the reef that are in good shape, rather, to areas that are in bad shape. So we can take an area with really low biomass on the left here and add coral back to it, so it becomes this vibrant ecosystem again. And we initially focus on restoring reef biomass. And that's our first priority because the reef, once the reef structure is restored, we start to see those ecosystem functions come back, like fish habitat, like shoreline protection. You can see all of these fish are very active in this reef with lots of coral cover where they can find microhabitats to live in, and not so many fish. We see one little yellow tang at this rubble zone. And we can engage in active restoration by a couple of different methods. I'm going to first start off by talking about more traditional restoration methods. And this is the primary focus of restoration in Florida, in the Florida Keys. In Florida, we focused on restoring corals by fragmenting corals that were already on the reef and planting them back out onto the reef. So you can see on the left, we've got small fragments of staghorn coral that are zip tied to nails, and they'll actually overgrow those nails, overgrow the reef, and join together to form these large colonies that can actually reproduce on their own. We can supplement these efforts through the use of land-based coral nurseries, such as the one that we are building outside. And the utility of these land-based nurseries is that we can fragment and grow corals under close supervision. It's kind of like, it's literally a nursery for these corals. We have people who have eyes on them. We can make sure they're not developing disease, and we can help them grow faster by feeding them. We can also use these land-based nurseries to grow really delicate coral babies. So we can engage in assisted reproduction on land, which I'll talk a little bit about, and then nurture those corals up on land until they're big enough to go back out onto the reef. One other practice that we engaged in successfully in Florida was the use of fragmentation and reskinning. So fragmentation, here we're cutting a coral up with a bandsaw, and this actually facilitates faster coral in growth by stimulating the coral to grow over their wounds. So think about if you cut your arm, your skin cells are turning over much more quickly on that wound than they would on the rest of your body. And this makes many copies of a single coral. So these are genetically identical plugs of the same coral. And when we outplant these back on the reef, we can reskin the reef by putting those clones next to each other, and they'll actually recognize each other and fuse together. So this is an example of a coral that an outplant array that led to a colony being produced that was sexually mature within 3 years, which is really important. In the Florida Keys, we were seeing there was no natural recruitment of this species, and very few sexually mature colonies were left. So the ability to get sexually mature colonies from our outplanting efforts was huge. And the reason creating these large colonies faster is so important is because coral sexual maturity is size dependent. Just super weird. Like imagine if taller people were the only ones that could reproduce. But it means that corals have to reach a certain size before they can sexually reproduce and create that next generation. So here you can see, a little hard to tell, but those were outplants at one point. You can see that circular shape. They fused together, and this colony was big enough that it could be sexually mature. And the reason outplanting identical coral works is because corals are this colonial animal that's made up of many individual polyps, like we talked about earlier. And each one of these polyps is genetically identical. So it doesn't know when you outplant a coral next to it, it starts growing against each other. They're like, oh, that's me. I'll fuse back together. If you put corals that are not identical next to each other, they'll actually fight and potentially kill each other. But we can harness this principle to engage in outplanting-based restoration. And we can outplant these clonal fragments that then recognize each other and fuse, and they reach those larger sizes faster. That allows us to reach sexual maturity. However, without thousands of people outplanting corals in this manner, achieving success at an ecologically relevant scale is really hard. I mean, we'd have to be putting these corals out at so many reefs. We need thousands of people. It also depends on these corals surviving for many years, despite these environmental stressors that we've been talking about. So if we're just putting corals out there and they get cooked the next year from a bleaching event, was it worth all that effort it took to grow them on land? And these stressors that corals are experiencing are projected to get worse, as I showed earlier, but this is a model of where ocean temperatures are expected to go. The temperature of our oceans has risen dramatically since the 80s, and there is no indication that this trajectory is slowing. So if we just outplanted clonal colonies with no consideration for how they'll respond to stressors, a genetically identical population or a monoculture can easily go extinct from a stress such as thermal stress. So if one particular genetic variety is really susceptible to heat stress, it can just die out. Whereas a genetically diverse population is more likely to have individuals that can survive that stress event and carry on the population. Think of how some of us are more resistant to COVID or the common cold. We can harness natural variation in the corals, in the coral population, to engage in restoration of a diverse population. And that genetic variation occurs naturally, and it's going to help these populations cope with rapid environmental change. So how do we create that new genetic diversity that's needed to respond to stressors? Coral is an animal. Just like all animals, corals sexually reproduce, and that introduces new genetic variants into the population. And most stony coral species reproduce by what's called broadcast spawning, where they a hermaphroditic colony, meaning it's simultaneously male and female. That single colony releases a bundle that has both eggs and sperm. And they release these bundles during annual mass synchronized spawning events that depend on the lunar cycles. So under normal conditions, you have many colonies releasing these sperm and eggs, and you have genetically distinct parent colonies that recombine. You have an egg that gets fertilized by the sperm from a different colony. This should look familiar from health class. It's the same principles that our sexual recombination follows. And normally this results in larvae that will settle down onto the reef through a process called recruitment. And these coral babies or sexual recruits are all genetically unique from their parents and siblings, just like humans are. So we've introduced new genetic variation through the process of sexual recombination, which shuffles genes, resulting in offspring that have different combinations of traits that they've inherited from their parents. And this random shuffling process of DNA is how we get genetic variation. So why has genetic diversity become a problem in places like Florida if coral sex is happening? The problem is sexual cycles can be failing in the wild, especially in places like Florida where coral cover is really low. We saw that picture of that essentially a coral that was a reef that was just a rubble zone in Florida, and we actually saw one in Hawaii as well. Coral eggs and sperm are only viable for a few hours after they've been released, which is kind of an error in and it's not a good genetic function, but they're only viable for a few hours. So in some cases, fertilization is failing because spawning's happening on different nights or at different times. So one colony spawns on Tuesday, the next one spawns on Wednesday. They're just not going to mix. And sometimes it fails because the communities are not dense enough. So those colonies that are reproducing are too far apart. So there's just not that mixing happening. And even if fertilization does happen, in some cases, the reef environment is not suitable for recruitment because it's taken over by turf and macroalgae, and that prevents the larvae from settling on the reef. So in some places you may have seen, thankfully we've done pretty well in Hawaii, and our parrotfish actually do really well at keeping our reefs from being colonized from algae. So this is one of the many, many reasons we need to maintain fish abundance and diversity on our reefs. These herbivores eat the algae that would otherwise prevent coral recruitment. So low genetic diversity is a problem in some areas of the United States reefs, especially in Florida, and it could become a problem in Hawaii without intervention. So we found ways that we can insert more genetic diversity into each step of the coral restoration cycle. Don't worry about reading this. I'm going to walk us through it. As I just mentioned, the best way to introduce more diversity into a population is through sexual reproduction. But sometimes corals have a hard time making babies in the wild. So we can assist with coral reproduction in two main ways. We can increase population density so that the corals find each other and those matches are made, or we can help corals, we can get hands-on and help them make babies in the lab. In terms of the first method, I showed you that outplanted corals can get big enough to make their own babies. And while I was at Moat in the Florida Keys, we saw the first of our outplanted staghorn populations to reach sexual maturity and spawn. So we finally were able to link that whole circle together. And the idea there is to get restored populations to a self-sustaining state, but we don't have to keep adding more density. Those corals are going to make babies on their own. And this circle here is a little hard for you all to see, but these are actually those egg bundles I talked about. So we can crack open a outplanted coral that is sexually mature and show that it's producing those egg and sperm bundles on its own. And this is the same coral releasing those egg and sperm bundles. So this is staghorn coral, Acropora cervicornis. And this coral spawns for about an hour around midnight for a week or so after the August full moon. And different species spawn during entirely different points of the year. So we don't have to worry too much about this species mixing with a different one. They've bioengineered that over millennia of evolution. And each one of these little heads here, sorry, it's a little bit dark. We filmed this at night, so a little tricky, but each of one of these protrusions here is a coral polyp, that tiny anemone we talked about. And in the center of that polyp is a mouth where it's releasing a bundle of egg and sperm. And that bundle floats to the surface because it's full of yummy lipids and fats, and that's going to help carry that bundle to the surface of the water, and those lipids will break apart and the eggs and sperm will dissipate. And that can actually it can allow the eggs and sperm to go to reefs that are pretty far away, as long as the ocean currents are strong enough that it can carry them in a few hours. But in Florida, we are not seeing the mixing of these bundles from one colony with other corals. So we were able to get hands-on with these corals and assist with coral reproduction by facilitating crosses. So we were able to bring these colonies on land and facilitate making matches between colonies that we're not normally mixing. And that gives the coral eggs a better chance to get fertilized, but it also allows us to make targeted crosses of parents that we know are strong. Like I said, we might know that one of us is really resistant to COVID and one of us is resistant to the common cold or grew really tall. If we bred those two parents together, we might get progeny that are better at both things. So this allows us to create new genetic variants that have potentially beneficial genes that can facilitate reef recovery. And importantly, this is stuff that would be happening on its own if the reef was better at making babies. If there was more coral density, all of these matches would occur. We're just helping the process along. So what this looks like in the lab is we have to collect these bundles at night. We don't use white light because it can confuse the corals about what time they're supposed to spawn. They think it's not nighttime. So we collected these gamete bundles by hand with pipettes. And as you can imagine, it's a little hard because, on one hand, it's hard because there's so many of them, but it also makes it's easy because they're, a dime a dozen. But if we're trying to make targeted crosses and we want to make sure that all of these matches are happening within the time that they're viable, it's a lot of work. It takes an army. Thankfully, we had lots of help, lots of volunteers who came and assisted us. And after we collected these bundles, we would wait for them to break up where the egg naturally separates from the sperm. And here you can see, I don't know how much my podium is blocking you, but this is the egg and sperm bundle as it starts to break up. So you can see the little pink eggs, and then inside that white cloud is the sperm. So this happens naturally. The lipid bilayer around the bundle dissolves and the eggs and sperm get released separately. But when we assist in this in the lab, we take eggs and sperm from different parents and mix them together. And these fertilized eggs then develop into swimming larvae, planula larvae. So this is a tiny little baby swimming coral. It's kind of crazy to think of our giant stoic corals being this little swimmer at one point. And we can harness this process to, we could step in and then release swimming larvae onto the reef. At this stage, we've helped make the crosses. We've given the babies a head start. So once those matches are made, we can then release the larvae onto the reef itself and let the reef take over. And this is an example of larval release happening at a large scale in Australia. So all of that pink stuff is fertilized eggs. So they've started to become swimming larvae. But we can also raise the corals on land in a protected nursery like the one we have outside. And we had a lot of success in this in Florida. We produced some very strong corals that I'll touch back on later. And once a larvae is swimming, it then metamorphoses to form a primary polyp, which here we're starting to see that anemone shape. We've got a ring of tentacles outside of a mouth. And I wish you could see it a little bit better, but these are corals as they start to deposit their skeletons and form new polyps. And what this guy looks like, a little hard to see, but this is their first rings of tentacles here. This looks just like an adult coral, except that it's got no color, right? It's still bleached. And it's because most corals are not born with that algae that gives it its food. So as the corals grow, they will start to uptake that symbiotic algae. So over the next few months of their life, we carefully care for them as they develop, as they start to grow more polyps. You can see here, and they're starting to uptake from the environment that single-celled algae that gives them their nutrition. So you can actually see, I don't know how well you can see, but there are tiny little dots here, and that's the algae starting to colonize the coral. And by the end of about 6 months, they have numerous polyps, and they've reached their full pigmentation. And at that stage, they're kind of weaned off the teat. They don't need to be fed. They are starting to produce nutrition on their own through photosynthesis. And as we grow up these babies on land and settle them onto plugs, we can also step in and sequence parts of the coral genome to figure out how closely related those babies that we're making might be. We kind of make a family tree of these coral because we want to make sure that we're not producing a whole bunch of siblings from one family. We want to make sure we're producing babies that are as genetically diverse as the people in this room, not just me and my 10 sisters. And that's to ensure all of these principles we've talked about, that sexual recombination keeps happening and that there's enough genetic diversity to respond to stressors. So assisted sexual reproduction is a huge tool. It increases genetic diversity and it can help scale up restoration efforts if we're releasing larvae at scale. But what if we're just breeding babies that can't take the heat? We know that there's natural stressors going on. So we can prevent these issues by integrating research at every step of the restoration pipeline. And my research is primarily on stress tolerance. So how can we breed and outplant corals that are more resistant to things like bleaching or sedimentation? And there is natural variation in all of these stress responses. And that's not only based on who the coral is, but what it's nature versus nurture. Corals that grew up in a more stressful area, such as an area with lots of sediment input, or maybe a shallow site that experienced a huge temperature range during the course of the day. Those corals can be acclimatized, they become acclimatized to those stressors, and they can actually be stronger in the face of future stressors. So it can be kind of intuitive. If you grew up in a bad household, maybe you've got some things you're not so good at, but you're really tough. You've learned to deal with some problems and you're stronger for it. And as I've discussed, natural variation in tolerance exists as well. Like how certain humans are more resistant to certain diseases. So if we can harness these naturally resilient corals, both nature and nurture, for reproduction, we can produce populations that are stronger. And just some indication of the natural genetic variation that's present. This is an elkhorn coral from the Florida Keys. And here we have two genetic variants that are right next to each other, experiencing the same conditions. And I know they look like they're fused, but as you will see, they respond very differently during a bleaching event. So one of the corals stayed very healthy during bleaching, and the other coral almost entirely bleached. It lost its nutritional support, and then it eventually died. So that coral on the right is now completely dead and will eventually start to degrade and break off of the reef. So we can harness this genetic diversity and make it, it's functionally equivalent to resilience. If we have lots of different genetic, different variants, we can assume that some of them are going to be stronger in the face of future stressors. But we can also measure resilience. So we can go in and see which corals are stronger against certain stressors. So we will conduct stress testing here to see which genets are better at dealing with different stressors. So here I'm showing an experiment that I ran in the Florida Keys, testing resistance to thermal stress. So we heated the corals up and saw which ones cooked and which ones didn't, which is so cool to see. There's natural resilience at play here. We have exposed to the same exact temperatures, one genetic variant stayed fairly pigmented, it retained a lot of its algae and didn't die. And then one of these genetic variants, you can see it's starting to get colonized by algae, and it did eventually croak. So we know that there's these naturally stress-resistant variants that exist that defy the odds in the face of these stressors. So we might think of these strong corals as supercorals. We see here, this is a picture from French Polynesia during that really nasty bleaching event. And there are some corals here, that same species, that are doing well. They're totally pigmented, even when the other ones are stark white. So these corals, maybe they're supercorals. Maybe they have something in them that makes them very strong against this specific stressor. But these stress-resistant variants are often really rare in a population. We can find a varying degree of how strong they are in response to low-level stressors, but when you cook the corals, it's going to be very rare to find corals that are entirely resistant to that stress. But what if we picked those hardy corals and used them for sexual reproduction? So, we picked that single rare coral that did really well, and we bred it with some other corals. So we could take that superpower and select for it. We select those temperature-tolerant variants that we've identified through work in our nursery, breed them together, and then in the next generation, we can actually see an improvement. So we've demonstrated this in Florida that we can enhance stress performance through assisted breeding, and that pushes population level in response to these stressors. And this is not a new concept. These concepts have been applied for millennia in agriculture. For example, it's not genetic modification, it's selecting for things like stem length or how juicy your fruit is. Farmers have done that for as long as agriculture has existed. And for example, in one of the longest genetics experiments done to date, farmers selected for corn that had a high oil content. So they really wanted those kernels to be nice and juicy. And they were able to breed very juicy corn together with other juicy corn. And they pushed the population oil content values from 5% to 20%. And that took 80 generations of breeding. So it wasn't fast, but it's a natural speed. Progress is slow, but we can assist this progress by helping to find those stronger strains. So if we know which genetic variants are more tolerant of certain stressors, we can use that info to make crosses that will produce stress tolerant and genetically diverse corals that we can use in restoration. So maybe we mix a coral that grows really fast with one that's more tolerant of high heat. And as I mentioned earlier, if sexual cycles weren't having a hard time in nature, all of these crosses would be already taking place. We're just harnessing existing natural variation and helping these crosses along that are beneficial. So in some sense, we do have the capability to breed corals that are super corals in the face of certain stressors. And I promise you this is a success story, even though it's another bleached coral. This is a parent colony that was used for sexual reproduction activity in the Florida Keys. This is a pretty substantial bleaching event we had last summer. You can see that it is as white as it can be, but this is its baby, that same year, that same day, in the same nursery. And you can actually see that maybe a little hard, but its polyps are out. That coral is happy, it's feeding, and that is a coral that we produced through sexual reproduction, breeding parents that were strong together. We bred parents that showed signs of bleaching resistance, and we produced progeny that was stronger. So these principles of selective breeding work can be broadly applied. We've shown they work in Florida, and it's something that we may engage in here as we develop our program. And there are lots of different traits that could be important. I've talked a lot about thermal stress. It's a big one. It's really rough on coral, but there's lots of different traits that are important. We might select for fast growers. We might select for good moms and dads. We might select for corals that have high disease resistance. We don't have a ton of disease in Hawaii, but there's a fair amount in Maui. So it's something that's something to consider. And maybe we breed corals that are high performers in a variety of traits to keep increasing those traits without selecting, maybe all of those bleaching tolerant ones are related. And if we keep breeding them, we might reduce the genetic diversity of our pool. So it's important to select for a diversity of traits. And the other factor is that all of these stressors are co-occurring. Thermal stress doesn't occur in isolation. It's occurring here in tandem with sedimentation, in tandem with potentially disease, in tandem with storm activity. So a high performer in one trait isn't necessarily good at everything. And especially with all of these stressors occurring at once, there's probably going to be some trade-offs. We think that there might be corals that are really, really good at one thing, but lousy at others. So it's unlikely we're going to find corals that are good at everything all at once, at least right now. So that's why it's so important to conduct restoration that's focused on diversity first and foremost. Yes, we should identify corals that are fast growing. Yes, we should identify corals that are bleaching tolerant. Get more of them out there, but also increase the amount of diversity we have on the reef and let the environment select. If we can, that same principle I showed you earlier, we don't want a monoculture. We want as much genetic diversity out there and let the environment select for it. And we can always add more diversity in through sexual reproduction as we start to lose genets that don't perform well. So I think of this like a diversified stock portfolio. Not that I have a stock portfolio, I'm 30. Oh, sorry. You want to invest in you want to put your money in multiple different forms to prevent against future problems. So, you might have some money under your bed, under your mattress. You might have some money in real estate. We want to engage in restoration that diversifies our options. We might pick some corals that are bleaching tolerant. We might pick some that are fast growers. We don't want to focus too much on any one trait. And we always want to maintain diversity because we just can't predict what the future reefs are going to look like. And when I was in the Florida Keys, I really focused a lot on these principles, and I developed a genetic management strategy for moat and for other coral restoration practitioners in Florida. And this was distilling these principles of diversity and resilience into a written document, a white paper that other practitioners could use to inform restoration practices. And some of these principles we will use to help develop the ʻākoʻakoʻa program as we engage in sexual recombination and reattachment and reintroduction of corals. One of the last things that we can think about is how larvae will naturally spread from currents. This is a map that we were able to make on how those babies are naturally spreading through ocean currents in the Florida Keys. So we were able to use ocean currents to predict where those rising gamete bundles were going to go. If the currents are fast, maybe they can get as far as moving from Key West all the way to St. Lucie on a really good day. And we can use these to inform where we engage in restoration. So if we see that lots of babies are being made in Key West that go all over the Florida Keys, we should really focus our restoration efforts on that source area. And we can start to make those maps for Hawaii as well. And this is already, this work has already begun here. This is a map of our ocean currents. And from these currents, my boss's group, Dr. Greg Asner, they were able to develop these larval connectivity maps. So predicting where larvae will move because of these ocean currents. So they simulated larval flow for different species and during different spawning windows. And we can then make inferences about not only where the larvae will end up, but where they started. So as I mentioned, that can be really important for finding areas to restore because those reefs are sources of babies for all of West Hawaii. And we can verify these models by putting out literal tiles at these reefs and seeing how many babies settle on those tiles. So we can make the models from ocean currents and verify them with field data. So to summarize, I know I've thrown a lot at you guys today. I'm obviously super excited about coral restoration, and we're still engaging in the planning stages here for the ʻākoʻakoʻa program. But the main areas of interest that we're going to be prioritizing are replicating those strong lineages that we identify on the reef. So if we identify very thermally tolerant or fast-growing lineages, we can grow them in our land-based nursery, replicate them into many fragments, and then put them directly out onto the reef. And that's work we can start engaging in very soon, as we can get corals into those tanks, which you can imagine, I'm very eager to get corals in. But we'll also work on reproducing genetically diverse and resilient lineages. So taking those strong parents that we've identified to produce genetically diverse offspring that can be restored either as babies that we raise up here or as larvae that we release onto the reef directly. And then the last thing that I haven't talked about too much today is that there's a lot of really great coral already out there. There's huge corals that have withstood all these different bleaching events that Hawaii has experienced. They've withstood storms, but maybe in the past few years, they've become eroded at their bottom and they've become a little top heavy and they've tilted over. We can get, with communication from all of the partners that we're engaged with, we could hear about these corals that have become dislodged, and we can get out there and literally cement them back out onto the reef. And the reason these big colonies are so important is, they've been resilient against years of stressors, and they're already spawning age. These big guys are already primed to make babies that are also strong. So it seems low tech, but just getting out there and putting the corals back where they came from is going to be really important. And then working to stabilize the coral rubble that exists. I showed you that picture of Hononau, where we've just got dead reef skeletons scattered across the place. We're working very closely with the Division of Aquatic Resources, who is piloting some methods to stabilize the rubble itself. If we can get those coral skeletons in a place where they're not tumbling, then we can get natural recruitment in that place again. Right now, if you have a baby coral settling on a finger coral that's just tumbling, it's just going to get smushed. So one of their priorities is to restabilize those rubble fields. And all of our work depends on 4 pillars that we are continuing to develop as we refine our program. But I've talked a lot about the science today. I'm obviously part of the restoration team, but we have a very strong diagnostics aspect to our science as well. And that involves, you can see some of the maps around you. We're able to go out and map the reefs and see where the problems are. We can identify areas of increased coral mortality. We can identify areas that are experiencing a lot of sedimentation or turbidity. And our team has been able to go out during the bleaching events in recent years and identify areas that have been hit the hardest that need restoration. We're also actively working to understand water quality issues on the island so that we can both stay involved with the government to change policies that might influence runoff, but also identify corals that might be resistant to these important compounds. So say we identify that there's a certain pesticide being released in an area, we can engage in multiple ways. We'll talk with the community, we engage with the government to prevent the release of that pesticide or what have you, and we can identify what the coral's limits are for that threshold, show quantitatively how much a coral can survive. And then all of our research is strongly dependent on our cultural engagement and the communities that we're working with. I'm not from Hawaii, I'm still very new, and I'm constantly learning what is important to Hawaiians and what reefs are most culturally relevant. So we have engaged a very robust cultural advisory group to develop our restorations built on a foundation of cultural knowledge. So we've partnered with community organizations that are focused on maintaining the island's cultural heritage and natural resources. And we want our plans to align with the needs and values of these communities that represent the entire coast of West Hawaii. And before we engage in any restoration efforts, we'll present our methods to these leaders and to other community members to make sure that we're doing what they want and what they feel is right. And we can refine our strategies as we receive feedback from these communities. So we're not operating in a vacuum. We want to hear from people like you and people from your communities about what you think is important, what reefs you see that are degrading, and making sure that what we are doing aligns with what you believe is right. And with that, I want to thank everyone that has been involved in this project. As I mentioned, I'm part of the ʻākoʻakoʻa restoration team based here, partner pretty heavily with the Division of Aquatic Resources from the Hawaii State Government, part of DLNR. I talked a lot about my work at Mote Marine Lab today, and amazing people who have built me up in my career. And that's it. I have no idea what time it is.
You just compressed a three-credit class.
Do you feel like you learned something?
I have at least one question per slide. Algae.
Mm-hmm.
What does the algae get out of it?
That's a really great question. Not a lot. They get a hospice, essentially. They get an area to live in. Otherwise, that algae is just bouncing around out in the ocean. And that algae, it can be free living, but it does a lot better when it's housed inside of something. So that algae is not going to be able to readily photosynthesize if it starts sinking to the pelagic level. We want to, that algae wants to receive a lot of light. So finding something it can stick onto makes that algae really happy. But it's something that we still don't understand very well. The algae gives a lot more to the coral than it gets. The algae does get some sugars back from the coral, so there's some extent of partnership, but a lot of it is just that safe place for the algae to rest.
Can you tell us more about what's going on over there? How big is it? Timelines, just anything?
Sure. Yeah. So it's going to be 72 raceways. All of them have been installed. We have 36 that are plumbed and running right now. We do not have coral in them yet. We're making sure that our water quality is appropriate for the corals. We still don't know how much it costs to run each raceway. We've been so great. We're so grateful to have the host park because they do all sorts of amazing testing on their water, which is sourced just down at the end of the host park there. So it's piped all the way up to us. And they are making sure that they're not providing water that's toxic to the corals. But on top of that, we're making sure that our dissolved oxygen is high enough, our pH is correct. We want to make sure the temperature is pretty consistent for those corals. So we are having our ribbon cutting ceremony next month. So once we are officially open, we will start putting corals in tanks and starting to ask some experimental questions. Some of our first questions are how fast each coral grows in our land-based nursery. Maybe some of these corals don't like being on land. Some corals really like high flow environments. So we've got a lot of questions before we start using the nursery for active fragmentation and outplanting. But it will be an immensely important tool for especially our sexual reproduction efforts because we can really care for any babies that we make here, and we can house those mom and dad colonies on land before they release all of their gametes. We can make sure that they're well fed and they have the right conditions. So we will be very large scale once everything is online. We're still still got a lot of dust, but the hope is that we'll be starting to put coral there in the next couple of months. I saw a hand back there first.
Yeah, I was curious, how much does the genetic makeup of the algae, the zooxanthellae, affect resilience versus the coral?
Absolutely, and I did not have time to talk about that today, but yes, absolutely, there's genetic variation within the algae. There are 4 main genera of symbiotic algae that they're genera that associate with corals. In this region, we see mostly the genus ʻākoʻakoʻa, which is considered a symbiotic generalist. It is not taking, it's giving a fair amount to the coral. It's not really taking anything it doesn't need. But there are variants of the symbiotic algae that are really stress tolerant, that are tolerant of those higher temperatures. But they're considered greedy symbionts. They take more of those sugars from the coral, and they can keep them alive longer during bleaching events, but they're not feeding them quite as well. So it's absolutely a factor, but it's very strongly driven by location. In some areas, we just don't find that thermally tolerant symbiont. And to an extent, it's driven by things like nutrient levels. The algae, just like plants, they really like being fertilized. So some variants prefer having a little bit more fertilizer than others. So it's something that we will be quantifying how much variation there is in the natural distribution of those algae. And it's something that you can harness as you make babies. You can introduce those different types of algae and into the babies as I showed you them slowly adding cells of algae and becoming more pigmented. So they can uptake different types. Often they don't keep them for very long. So it's an active field of development in the entire industry, making sure that those new symbiont strains are kept by the coral. Yep.
Where in the harvesting and specific species?
So we're working with several different species. We've got at least 5 priority species right now, 3 species of Parietes, and then 2 species of cauliflower coral. And our plans right now are not to move coral between embayments. We want to keep each coral within each ʻākoʻakoʻa that they're harvested from, at least, for the time being, making sure that we're not moving coral too far. And that's both because these ʻākoʻakoʻa have been shepherded for generations. So those coral they're endemic to that region. But an additional consideration is there's a potential for introducing pests if we move corals from different areas. So it's something that's on our minds because, as I mentioned, there's regions that if they've experienced more stress, they might be more tolerant. So, for example, if we harvest corals from Pu'ako, which has experienced a lot of land inputs over the years, those corals might be more acclimated to certain stressors. In theory, if we move those elsewhere, they could be stronger, but it's something that we're years away from doing. At this stage, we're keeping corals in the regions that they're from, and we really want to maintain the local communities that are from each site. We don't know yet. We have a couple of areas that we're prioritizing initially. DAR is working heavily in ʻākoʻakoʻa, and we're probably going to prioritize Hononau and Ka'alu'u initially, but that's something that we need to develop a plan with the community. So it's dependent on the reefs that are the most degraded and areas that we have source corals from. So if the whole reef is beat to heck and there's no source corals that we can move, that reef might have to wait until we are able to move corals between areas. But that's something that we're actively developing and start seeking out funding for. Yep.
Cauliflower corals come in several different colors.
Mm-hmm.
We see tan ones, lavender ones, green ones, something in between. What's the cause of that variation?
So it's primarily a result of the algae, different balances of, as I mentioned, there are 4 genera of algae. So different balances of those algae can change the pigmentation, but it's also dependent on the host itself. So on the coral itself. So they can secrete a tissue layer that is slightly more pigmented, more melanin, for example. So we don't know exactly what causes those different pigments, but it's a really good way to identify different genetic variants in the wild. So if we see that pink variant of cauliflower coral versus a tan one, we can be pretty confident that those are genetically distinct. But it's similar to us having different skin colors. There's all sorts of accessory pigments that are involved in that coloration, plus the algae changing that shade as well.
So, one follow-up.
Mm-hmm.
I've heard it suggested that corals are actually evolving in color.
Yeah, so there's been some evidence of that in the Great Barrier Reef that the brighter, the thing is the darker ones may have more photoprotection. If they've got a darker tissue, it may protect their symbionts more from heat stress and also light stress, because light stress also stresses corals, as it turns out. But some corals have evolved to be different colors to recruit more algae. We think that there's certain pigments that may be cues for algae. But the corals that I've worked with for a long time in Florida, they're all brown for the most part. We've got a little bit of green, but we still don't exactly know what causes those colors, although we can say pretty confidently that green is a result of green fluorescent protein. We can put it under UV light and it glows really nicely. And in Australia, during bleaching events, there's been a lot of, it's been highly visible that the corals turn these beautiful UV colors when they bleach. And that's not a method of adaptation, that's kind of a cry for help. They produce more fluorescent proteins as they've lost their symbionts. We don't know why. It may provide some photo protection, but it's still something that's being actively explored. It's crazy for organisms that have been around for so long. There's so many questions we still have about their basic biology. We still don't exactly know how they kick out their algae during bleaching. We have some really good ideas, but it varies by species, and it's hard to quantify. So these are really great questions that I wish I had better answers for.
When you plant colonies you've cultivated, you have to actually manually place them. You can't just broadcast them to the air.
No, not adult colonies. The larvae, absolutely. And that's one of the things that makes larval release so scalable, is because that larvae is, its next step in life is to settle on something and stick. You absolutely can just release the larvae. Of course, you want to monitor it and make sure that it's sticking. You can use settlement tiles. But no, adult corals, we can't just, for the most part, we can't just chuck them off the back of a reef. I'm not sure we'd get, or the back of a boat. Don't think we get permits for that. Some coral species, in theory, you could do that if the wave energy was low enough. So staghorn coral in Florida grows really quickly, and as long as it can get lodged somewhere in the reef, it can grow. But here we have so much wave action that it's really unlikely if we just chucked coral off a boat, it's just going to keep getting tumbled and tumbled. So as I mentioned earlier in the talk, direct outplanting it's a lot of labor, and it's one of the many tools in our toolkit, but it can be really hard to scale it. But you have to, yeah. What we do is we actually epoxy or cement each of those corals down to the reef, and we've identified different types of epoxy that the coral doesn't mind, and it'll just grow right over it. And for certain species, you can hammer a nail into the reef and zip tie the coral to it. But as we started to find out here, our reefs are too hard. There's a lot of basalt here, so you're going to lose your arms if you try to hammer nails into it. Soccer Trina?
Are you guys going to do any volunteer days? I know you probably ways off.
That's a great question that Tater can answer, probably, and Andrea in the back.
As far as volunteer days, there's definitely on our website. I'm also with ʻākoʻakoʻa, if you didn't know, and Moamakai. On our website, there's a volunteer place where you can sign up, and that program is in the works as well. So yes, I would say you can also become a reef guardian, and we are hiring. So check out our website at ʻākoʻakoʻa. There is a place to sign up for volunteering.
And Cater's been helping to develop a vocational program where we can get especially young people trained in the skills that it takes to be involved in coral aquaculture. And as it turns out, there's a lot of different skills. It's not, you don't have to have a PhD to do it. I do one very specific thing. There's so much of it that's about plumbing, that's about caring for the corals, feeding them, getting into the field, getting scuba diving. So there's so many, boat handling, so many different skills that are important. And we really want to get people who are from these communities involved in these restoration efforts.
Do you have a timeline of when the volunteer activities might be?
I don't. I wish I did. Just keep checking back on our website. That's the
This
best
program
way.
is still very new, so stay tuned.
And
there's a mailing list.
Yeah, if you had signed up for the ʻākoʻakoʻa newsletter, then we'll have announcements for volunteer opportunities and ways you can help.
Yep.
Peter knows this, but Grace, if you want, we can get the local commercial recreational scuba industry involved, because we're out on the water all the time.
That's your guy to talk to in the floral blue with the hat. He works for DAR. They are very actively seeking divers. Not to put him on the spot. But yes, absolutely. Yeah. Yeah.
And yeah, because we're out on the water all the time.
Yeah. Well, Chris knows that. Yeah. Amazing. Yeah, and it's a platform that's worked really well in other areas, is getting dive shops involved in coral restoration. And we don't want to do restoration that's catered towards tourism at all, but in some places it's worked really well to get these visiting divers engaged in some of the simpler methods.
What we've seen recently is there's so much coral growing on the mooring lines itself.
Absolutely.
The buoys are actually sinking.
Right.
It's like you don't want to break it off to keep the buoy, right? But if the buoy sinks, then you have to drop an anchor, which
Yeah, so that's one of the
Chris knows this, right?
Yeah.
And add to that.
they've already, you know, it's on their docket.
Like you can't just drop it on the top.
Right. And then the really neat relationship of ASU of Arizona State with DAR is that we can, you know, bring in some of those corals from the mooring lines, identify which ones might be strong against certain stressors. And we don't have to just take that entire colony and direct outplant it. We can cut it in half, put some straight back onto the reef and bring some here and figure out what makes it tick. So Arizona State is going to be engaged in those active restoration activities very soon, but DAR is already very hands-on with those activities, dealing with things like mooring lines, dealing with boat groundings, and they're out on the boats every day that they can be.
And I'll just add to that, there is absolute communication between Malamakai Foundation, that works with the day-use moorings, and ʻākoʻakoʻa and DLNR. So we're all working for the same goal.
What's the thinking on introducing non-native species that have demonstrated resilience? Is that something we just
Probably not, because it's so hard to know whether it could become invasive, and it's a crazy thought to think that like you could introduce a coral and it would become invasive because we so desperately need coral. But the Caribbean's not doing it, and the Caribbean's in much worse straits than we are. So we will, you know, in some ways we are 10 years behind the Caribbean, at least in terms of how deteriorated our reefs are. So we can take those kind of desperate measures, be guided by interventions in Florida, and they're not yet introducing invasive species, well, non-native species, exactly for that reason. And it's not just whether that individual coral could spread physically, it's they start releasing their gametes, and then they're recombining and potentially genetically swamping our native corals. So unlikely, there is a patch of ʻākoʻakoʻa somewhere on the islands that we think is introduced, and it's something that we're hoping to test genetically and confirm that it is something that is introduced, or identify whether that's natural larval transport from a different area. It's entirely possible. My guess is if they're all clones, if they're all the exact same individual, that is something introduced. So great question. I would say at this time it's a hard no. Yep.
What are they doing to stabilize the rubble fluids? What are the types of things that they're doing?
Not to speak for the person in the room who knows more about this, but DAR is pretty much leading these efforts at this time on West Hawaii. And they're involved in a project with NOAA to right now get, there's a lot of clearance that has to be, you have to get those activities cleared with NOAA, with the Army Corps of Engineers. You have to make sure that you're not engaging in those activities on historically important sites. So they're right now involved in the permitting process for that. But after that point, they are intending to pilot a mesh. They're going to try a mesh that's made of stainless steel and a mesh that's made of extruded basalt, so basically lava rock, and adhering it to the reef with stainless steel rods to tamp down that coral rubble. You know, stainless steel is not the preference because it's not really a natural material, but if we can get that basalt mesh to be cost effective, that would be great. I think we've got a lot of evidence that would be, it's a natural material, it should be well accepted by the reef, but stainless steel is certainly a tool that's used a lot in the marine environment. It's not harmful, not significantly harmful. And just tamping down that rubble can help increase natural coral recruitment on that area. So we're all ears if you're thinking of other strategies. It really hasn't, on the steep reef slopes that we're working with in West Hawaii, nothing really has been done like that before. There's been rubble stabilization efforts in Puerto Rico, for example, but it's in reef flats. So it's much easier. We're working with rubble stabilization on areas that are sloped. So it's really a new frontier. But before we engage in those methods, you know, we're really talking to the community and making sure that that's something they want to see, regardless of, you know, whether it can impact recruitment.
What are the mooring lines made of? Because they obviously host coral.
The mooring lines, for the most part, are rope. Yeah. They're mostly rope, right? Yeah. So it's a natural fiber. But you definitely, you know, stainless steel is not a great thing for coral to settle on because it's the main problem is it's non-porous. So something needs to be there for the coral to hook onto. But you certainly see corals settling in the vicinity of stainless steel. And after it's been in the environment for so long, it's probably a little bit less toxic, but it's absolutely a question that we need to ask. And we're also not using stainless steel in a closed system. Any things that are being released from that are being moved very quickly around the ocean, but it's absolutely a worthwhile question whether there's toxicity effects. Do we have questions online?
We don't. That's
Cool.
what I'm standing up here.
All right, if there are no other questions, cut y'all loose. This has been Coral Restoration 101 with Dr. Klinges.